So now that we've talked a little bit about cells and what you can see in cells, now we're going to talk about how you might see cells and how you might study these things. So biologists use a tool called a microscope in order to actually see cells, and it's really how we investigate the biological worlds. So a little bit about the history of the microscope. Microscopes have actually been around for a very long time. Not what you might think about a microscope though. 1590 was actually the very first microscope that we classify. It was called the compound microscope. You can see it actually looks a little bit more like a telescope than anything. Just for the record, in class, we actually still use compound microscopes as well. Doesn't look like this, but the idea behind the compound microscope is actually very similar from 1590 to what it is now. A little bit about the vocabulary I'm going to be talking about. So in a microscope image, when you actually get your microscope focused, there's two different things we talk about. We talk about magnification. Now, magnification allows you to increase an object's apparent size. This is good because based off the cell demo before, cells are teeny tiny. So the whole point of the microscope is allow us to actually increase the size of the apparent size of the cells, so we can actually zoom in and see what's going on at a cellular level. So magnification, very good. Another thing that's very important though is resolution. So resolution is the actual clarity of the details. So have you ever zoomed in on an image that wasn't meant to be zoomed in on and it got really blurry, right? So that had magnification, but it didn't have resolution. So it's no good for us to zoom in on something if we can't see the details. So in a microscope image, it's really important to have both magnification and resolution. A lot of our high-powered microscopes allow us to have really great magnification while still maintaining resolution. We're going to look at the different types of microscopes that are out there. In class, we use a compound microscope. It's the first type of microscope that was found, and it's the most widely used. The reason it's most widely used is that it's typically inexpensive to make when comparing it to the other types of microscopes we're going to talk in class. So in the school, we actually don't have anything but a compound light microscope. It's what we can afford. Does the trick though. It can magnify image up to 2,000 times. So it allows us to zoom into really anything that we need to see at our level of science. How this works, and we're going to cover this actually in a little bit more detail when we talk about physics. Light is going to pass through two lenses. So there's going to be a lens at the top that you look through. There's also a lens at the bottom. By the light passing through those two lenses, the image becomes magnified in a nice clear manner that allows us to see. So this is what something might look like underneath a compound microscope. So you can see you've still got details and it's zoomed in quite a bit so we can see what's happening at a cellular level. Another type of microscope is this idea of an electron microscope. It is used to measure very small objects, often objects that are subcellular, so objects that are within the cell. DNA is like very skinny, tiny strands. We're going to talk about in mitosis a bit. It's within the cell's nucleus. They can also be used to study other types of or parts of the cells and viruses, which are also very small. Rather than having light passing through lenses, it's using beams of electrons. So it's not having light to refract through lenses, it's actually using beams of electrons in order to create the image. This makes it much more powerful. It also makes it much more expensive, which is why we don't have them in our schools. So this here is an example of an electron microscope image. So you can see the incredible amount of detail in this compared to our compound microscope picture. Another type of microscope is our transmission electron microscope. These are similar to electron microscopes. They're just a little bit more powerful. They can magnify up to 250,000 times. So remember, the compound microscope was only 2,000 times. This is 250,000 times, so quite a bit more detail. And if you're looking at something, this is actually zooming in on part of a cell organelle. So not only is it looking at a cell, not only can you see a cell organelle, it's zooming in on a small portion of a part of a cell organelle. So it's pretty great for the magnification end of things. Last type of microscope is the scanning electron microscope. So again, using electrons, it's got a really nice balance of magnification and resolution. It can magnify not quite as much as a transmission, but it can magnify up to 100,000 times. So again, still much more than the 2,000 times of the compound microscope. We're now going to look at the different parts of the microscope because when I use terms in class about a microscope, you need to be able to know what I'm talking about. So the arm is actually used to support the microscope when it's carried. The coarse adjustment knob is a very large knob on the side. It moves the actual stage of the microscope up and down. The fine adjustment knob moves the stage slightly, so it allows you to sharpen the image. You always want to adjust the coarse adjustment knob first, then you want to adjust the fine adjustment knob second. The diaphragm actually allows light in, so it regulates the amount of light into a specimen. The base helps to support the microscope. The light source is where the light actually comes from. The image itself that we create in class is coming from light passing through lenses. Therefore, if you don't have any light, you don't have an image. The stage helps to support the slide, and the stage clips help to hold the slide in place. We have a couple different parts here. We have something that's called the objective lens. The objective lens comes off the nose piece, and it magnifies anywhere from 10 to 40 times. The nose piece helps to hold the high and low power objective lenses and can be rotated, so it's what you're actually going to turn to change the magnification. And the eyepiece has the ocular lens. So again, we have two different lenses, the objective lens, and then we also have the ocular lens down here. So let's actually see what that looks like in terms of the microscope itself. Let's zoom out a little bit. So this is a microscope. So again, the base is just there to support the microscope, and the arm is second support to allow you to carry it. The light source is here, and you would flip it on using the switch on the side. The diaphragm is again what adjusts how much light enters. So light is coming up from here. The diaphragm you can make wider or smaller if you want your image brighter or darker. The coarse adjustment knob is this big knob here. You're actually going to use this to move the stage up and down. So the stage will move closer or further away from the lens. This will help to clarify your image. The fine adjustment knob is right here. You're going to use it for just tiny minor tweaks after you get the coarse adjustment knob. The stage clips lift on the stage. So you can see we have a slide that we slide right into the stage clips. Oftentimes, there's very tiny subtle movements in the slide that translate to really big movements for what you're trying to look to. So the idea is you don't want your slide to move at all, especially once you get it focused. So the stage clips prevent it from moving once you get it focused. This nose piece here has our different objective lenses. So we have a 4 times lens, a 10 times lens, and a 40 times lens. So we have low power for the 4, medium is the 10, and high is the 40. And then we have the body tube up here that holds the ocular lens. The ocular lens is on the end, and the ocular lens has a magnification of 10. So you're always doing 10 times your objective lens. So for example, if you are on low power 4 times, it'd be 10 times 4. If you're on medium, so 10 times 10 times 10, which is 100. And then if you're on the highest power, that's 40. So you do 10 times 40, which is 400. Just in terms of safety, whenever you store the microscope, you always want to store it on low power because here, this lens is very expensive. It's made of glass. And this down here, the end of the diaphragm, also made of glass. So if you store these together, if the microscope gets bumped, there's a chance you could break the lens. So you always want to store it on low power. You also want to be very careful that when you're using the coarse adjustment knob, you only use the coarse adjustment knob on low and medium power. Again, on high power, the lens is very close to the stage. There's a chance that the coarse adjustment knob could swing the stage into the lens and you could break it. So that's the parts of the microscope. Next, we're going to walk you through this concept of scientific drawings. So I'm just going to highlight a couple things. I'm assuming you can read this on your own, but these are a couple rules that I want to go through that people make mistakes with scientific drawings. So scientific drawings have specific types of rules, and it's what we do. They're the drawings that we use to communicate what we see in a microscope. So I always say you don't have to be a good artist to be a great scientific drawings. You just have to be very careful and you have to be very precise. So you want to make sure that you examine the significant features. You don't just want to sketch the outside of a leaf. Is there more important detail in there that you need to include? Draw only what you see. If you don't see it, don't include it. So if you don't see a nucleus, don't include that nucleus because you think it's going to make me happy. This is really important, pencil only. So students always try to draw in ink. I will not accept diagrams in ink. So please make sure that your diagrams are pencil only. They have to be large. So oftentimes students will try to cram like 8 diagrams on one page. A diagram should be no smaller than a half a page. Usually scientific drawings, we do a full page. Sometimes I say save the trees and you can go ahead and do a half a page. No smaller than a half a page because then it's lacking the detail that's required. And you always want to use distinct lines, not sketching. So oftentimes people think they're artists and they do these beautiful little sketch marks that looks like this. That's wrong. You want to make sure you have a distinct line that shows the outside. When you want to show darker areas, so let's say there's like a really dark center or there's an area that's darker than another, you are not allowed to color. You are only allowed to draw lines and dots. So in order to show darker areas, you're going to do what's called stippling. So stippling is you actually do a bunch of little dots together, and that's going to show the illusion of darkness. So here you can see that someone's actually colored these in. Here you can see someone's used stipples or dots to actually go through and show the darker areas. A little bit about labeling. So when you actually label your diagram, you want to make sure that you label using straight lines and put everything on the right. Do not label on the right or on the left-hand side. Also, when you label, you want to make sure that you use these nice straight lines. You don't want to have any lines crisscrossing. We have nice straight lines with the ruler off to the right-hand side with labels. You also want to make sure that you indicate the title. So you want to have the title of what you're looking at the top and underlined. And you always want to make sure that you have the magnification in the bottom right-hand corner. So that's really important. A lot of people miss that. So always put the magnification in the bottom right-hand corner. Let's say your magnification was 400 times. You would show that 400x. Outside of that, this just goes through a nice little overview of what I had there. So if you know how to properly use the microscope and you know how to properly draw things under the microscope, I would say we are ready to go ahead and start our first cell lab.